Also spotlight. Okay. And we're going to pick up today with the problem that we stopped on last time. So you had these messenger RNA sequence to then look up and see on this chart what are the amino acids that come from the messenger RNA sequence. So what did you do with this? Tell me the amino acids and just give me your best shot at pronouncing them. What do you got for the first? GCU.
Alanine.
Alanine, good. Is that right? That looks weird to me. Anyway, UUA, what would that be? Must be a tough one to pronounce.
Leucine.
Leucine? Yeah. Leucine. GCA. Alanine again. AUG, this is one I have memorized, just from my past. Methionine, that's the start codon that's always at the beginning of a sequence. And then GCU, we have alanine again. So that would give us the amino acids. And then this particular set, I'm going to put some dots at either end, because this would be part of a larger protein. So I will give you like 5 or 6 sets of bases on the exam for you to do this kind of problem, but a real protein would be a lot bigger. So this would be a little stretch in between.
Are you asking?
Yeah.
Okay, so is it just going to go around in a circle like that? So like we go to alanine, leucine, alanine.
No, there would be other ones out here. So this is like a little snapshot of a larger sequence. But these amino acids would then fold up into a 3D shape of a protein. So I want to go forward a couple slides and there's a certain picture I want to show that relates to, okay, this one right here. So during translation, that's actually what we did here. We're translating this messenger RNA sequence into this set of amino acids. And we're acting like we're a ribosome that does that. And you can see down here, this ribosome is reading the messenger RNA and it's producing amino acids here, like alanine, leucine, methionine, and so on. And that makes a protein chain. And this protein chain is going to fold up into a 3D shape of some kind of protein that does something for the cell. So you get a 3D shape, and the 3D shape of the protein is going to allow some kind of behavior or some kind of function for the cell. And what the protein does is dependent on the shape. And what the shape is depends on the amino acid sequence. And that depends eventually going back all the way to the DNA sequence. Here's why that's important. If we have in the DNA sequence a change in the DNA sequence, that's what we call a mutation, a change in the sequence. So if the sequence of DNA changes, that can change the sequence that you get in the messenger RNA. and that might change the amino acids, and that might change the shape of the protein and then its function. The function is what we actually care about. So what this means for microbes and for human health is that mutations, changes in the DNA, might change the behavior of the microbe by changing the messenger RNA, changing the amino acids, changing the protein shape, changing it, how it folds, how it behaves. And most of the time, the mutation is going to make the behavior worse. Like most of the time, mutations are going to make the protein in the end have a worse function. We'll say 99.999% of the time. But once in a while, it's possible that you get a mutation that changes DNA, changes messenger RNA, changes the protein, and makes it work better. It's just really, really rare. .001% of the time. This is when we care. This is when we care about a mutation that affects human health. That rare chance that you get a mutation that makes the microbe work better and make it more effective as a pathogen, make it more dangerous to us. So when we hear about in the news lately, Delta variant of the coronavirus, it's a change in the virus's genome that changed the messenger RNA, that changed the protein, that changed the behavior of that coronavirus, and it's better at infecting us. So good for the virus, bad for us. It's rare, but when you get a mutation that's better, that's going to give that virus or that bacterium, it's got the advantage. So last time in your notes, we wrote that if you've got a gene that gives you an advantage, that's going to make you outcompete your neighbors. That's when we care about mutations in bacteria and viruses that affect people's health. So that's kind of a preview of something that we're going to talk about with our virus section. How do you get mutants? What makes some of them better than others? What gives them a competitive advantage? But it's always going to come from their genome. What makes them better? Yeah.
So is the 0.01% like when they're naturally occurring, or is it
all of these would, well, we can make pathogens better in a lab. And you may have heard there's some theories that coronavirus was engineered in the lab to make it better as a pathogen. That can be done, but what I'm going to talk about in this class are the natural occurring mutations. Is that what you're asking?
Well, yeah, that's how, because like, I wasn't trying to be political.
No, I don't.
Yeah.
It's totally, like my mom asked me a version of that question because of something that she heard about.
So if
Yeah.
if it was like not naturally occurring and it was like created in the lab, would it be easier for it to have a successful mutation that would make it?
So if we decide to do purposeful mutation to make something better in the lab, like people are smart, and you would assume that they would do it correctly. You know what I mean?
Yeah, that's what I was asking.
Yeah.
So like it wouldn't be 0.01% if.
Yeah, if you were doing it on purpose in a lab, you usually are going to have like a much higher chance of like, yeah, I think that this would make it better. Yeah, but in a natural situation, it happens all the time, even if it's at a low rate, because once you get that better one, it's like super successful and it goes and infects more people and then you have more of it out in nature. So we're going to come back to this better, worse thing, but I want to give you another example of what if we changed something in this sequence. That's something I ask you to do on the exam. Like you have a question like this, and there's a certain sequence, and then I say, what if there was this change? Work it all out and see what happens to the protein. So I want to do an example of that, and then we'll go on and get into our virus section. But this concept of mutation making something worse, making something better is a really big deal for microbiology and human health. So I'm going to try to leave this section up here, but just change the rows. Change what we're starting with here. And do a few examples. Okay, so always in the DNA, we're going to start with the template strand. And I'm going to try to make it the same way that it was at the beginning. See if I can get this right off of my little note. Actually, somebody just read me the template strand that we had last time.
CGA, AAC, CGT, TAC.
Thank you. Is that it?
No.
One more.
It's CGA.
CGA. Okay. So that's a template strand of DNA. And on these test or practice questions, I always give you the template strand. And then you'll fill in the coding strand, and then the messenger RNA, and then the amino acids. On the exam, I give you a printout of this. I actually have like 12 of them in my office right now from previous years, and I'll use those and print out some more. You'll have a printout of this. So the kind of thing that we want to know about this is if I made a change someplace in the DNA, like for example, I changed the C right there. So let me leave it and indicate where the change is. That C is going to change to an A. That's what a mutation is. You got a change in the DNA sequence. So then what I would ask you to do is, all right, if that's the mutation, fill in the coding strand, fill in the messenger RNA, fill in the amino acids, and then see what's the ending change in the protein, if that was the mutation that we had. So give that a try. If that C changed to an A, and then I'll give you also a second mutation scenario that's separate from that one. The next one is, what if we had a G base inserting or adding in right here? What would that then do? So take a few minutes and work on those two problems. They're two separate things. Never know how long things like this should take. I also hate to feel rushed myself. Okay, so I don't see too many more pencils going. So what do you have for complementary DNA on the first one where this C is changed into A? What we got here? G, C, T, 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 A, G, C, A, T, T, thank you. G, C, T. Okay. How do you get the messenger RNA sequence now? Replace T with U, but which one of these do you use to get the sequence that you want? You're going to use the coding sequence to develop the messenger RNA sequence, except you replace T with a U. G, C, A, U, U, G, C, U. Did I do that right? Okay, so what amino acid here? That one ought to be the same, right? Alanine. Leucine, right? Is that the right? This one would be the same. Again, alanine. Did this one change? What is it now? Isoleucine. And this one would be the same, which was leucine again. Sorry, alanine. Thank you. Alanine. Okay, so we've had a change in one of the amino acids in the protein, and that might change how it folds. And that might change how the protein works. It's probably not going to make it better. It's probably going to make it worse. But that's kind of how you go through that process. The next one is a little bit more difficult, the G insertion. So let's go through that one. Okay, so what this is really doing is we've now got an extra G in there, coming in between those two bases. So that's going to give us G, C, T, 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 A, G, C, C. And now this T is like, almost like extra. T, sorry, it would be A, T, G, G, C, then we got this extra T hanging out there. Is that right? Is that what you got? Okay. So our messenger RNA, we're going to make that the same as the coding strand. We just replace the T's with U's. GCU, UU, A, GCCAAU, CCG, and then a U hanging out. Seem all right? Does that match what you got?
I know the CCGs.
This one?
It didn't change.
That's right, I did that one wrong. That's why I asked you guys to check. Yes, thank you. That's why I don't like to ask people to come up and write stuff. I think it's hard to write, talk at the same time. Okay, so this is still alanine, this is still leucine. Then we might have some changes. So what's the GCC amino acid? Still an alanine. What's AAU now? Asparagine. You know how when you eat asparagus and then you pee and it smells like that? That's what the smell comes from. They named that amino acid after asparagus. GGC, what's that one? Glycine. What's this one? You don't know enough. So there would be some more sequence coming after this. More sequence at the beginning. Now we've got two big changes. And also from here, there would be even more changes because you shifted the sets of three. That's called frame shift. But what it's done is it's made a really big change in the protein, and it's probably going to make it work worse. Some mutations have more of an effect than others. Yeah.
So when they do like going back kind of like the lab stuff, they just randomly pick a protein and try to fit in there, and they have like background knowledge to see.
So when I was in graduate school, what my part of my project was to take a bacterial weapon protein and try to figure out what it actually does, because back then we didn't know what that one did. And so what I did to it was I made a bunch of random mutations, like 100 versions, and then I tested them on the plant to see, is it making the plant more sick or less or what? And I could figure out which ones are the important ones. So to make something on purpose work better, you need to know exactly what it's doing. And you need to know what parts of the protein are the most important for what it binds. So like spike protein of Delta virus binding to human receptor, you would need to know how they bind each other and what are the important parts and try to know enough biochemistry to make it bind better. But in a natural situation, the mutations are going to come from some different sources. So they could come from UV light that damages DNA and makes mutations. That's why tanning gives you cancer sometimes. Natural mutations can come from environmental chemicals as well, but the big one where natural mutations come from is genome copying errors. Genome copying errors. Like when you make new RNA or when you copy DNA, the enzymes that do that make a mistake like 1 in a million. Or in humans, well, in humans it's about 1 in a million. In bacteria, it's about 1 in 100,000. And in viruses, it's about 1 in 1,000. They just have messed up DNA or genome copying. If this is kind of seeming like I don't get it or it's a struggle, like let me know that later and we could meet and go through practice problems, as many as you want. But this is how mutations end up making things worse or better. It's a DNA problem. It may or may not change the individual amino acids in the protein. So we're going to now switch slide sets, I think. Oh, there's, yeah. The same stuff that I just wrote up there. But if you all have this in the slides, right? Yeah. Same set of stuff. This slide you can cross off. That one I'm going to leave up and come back to it at a certain point after we get into viruses. When I was in college, my favorite class that I ever had was a class all about viruses. I really didn't know much about them before that class. And the teacher was pretty old, and she was really old school, and she just sat down at the old crank projector, and she would just write notes and draw, and it would go up on the screen, and there were no cartoons, no pictures, just her drawing stuff, but she was amazing. And ever since then, I have been just pretty crazy about viruses and how they act. I think that they are so interesting. What do if virus wanted something in their non-life because they're not alive? What's their goal? What makes something the best virus ever? What? To infect? Pretty much. So if viruses have a goal, what would make you the best virus in the world is to make as many more viruses as possible. Last year in King 1000, McKinley and Marley were in my class, and it was about viruses. So a lot of this stuff is going to be reviewed to you guys, but the best virus in the world is not the one that kills the most people. It's not the one that makes the most dramatic symptoms. It's the one that makes as many more viruses as possible. And coronavirus is the total winner of that. As far as human history, there have never been more copies of a human virus that existed on Earth than right now of how many coronaviruses exist amongst people. It's crazy. So not to be the most deadly, to make the most more of yourself. That makes you a successful virus. There are certain things out of this chart that I want you to kind of highlight, and this is too wordy. So I think what I want to do is write some separate notes that are about some of these, but in simpler words, about what viruses are like. So we know that they are not living. They are not considered alive because they don't do their own metabolism. They require a host cell to complete what we're going to call their life cycle. So they're not alive, but we say that they have a life cycle with steps because we don't know what else to call that. A virus cannot copy itself without getting into a host cell. They have to have a host cell. Here's another important cool fact. all life forms have viruses that infect them. So we tend to focus the most on the human-centric ones, viruses that make us sick. Plants can have viruses. Dogs can have viruses. Worms, fungus, even bacteria have viruses that infect them and kill them. That's just cool to think about. They are tiny. And then the last thing I want to write down specifically out of this list is that viruses are very specific to certain host cells. Not every virus can infect every organism in the world. Who has a dog? What kind of shots does your dog have to get?
Rabies.
Rabies? Like, I only know 2 because we don't have pets because we're Asian household. So you get rabies, your dog has to get rabies shots. And there's one called parvo. You ever heard of that? We had a puppy when I was a kid that was real little, and my parents were busy, and they didn't vaccinate it, and it caught parvo virus, and it died from it, and it was really sad. Like, they get tremors, and then they get paralyzed, and then they die. It's terrible, but no humans ever worry about parvo. We can't get it. That virus does not infect us. But rabies could infect a dog and us. I don't worry about getting a plant virus. That's not going to affect my life. I don't worry about getting a virus that kills bacteria. It can't infect me. So this host specificity is a big deal with viruses. This other stuff we'll cover as we come to separate slides. This picture is showing you kind of some sizes of viruses compared to bacteria. So yesterday in lab, when we were looking at Proteus, it's about this size. And Staphylococcus bacteria are about this size. And everybody was like, I can't find it on my graph slide. It's so hard. They're so small. We don't bother to try to look at viruses under the microscope because there's a couple of like giant ones. But most of them are so small that you can't even see them on a light microscope like we have. So they're really small. You can't see them with a normal microscope. Viruses tend to have these shape categories. So they're not alive, but they're particles, and they have particular shapes or shape categories. Which are called helical. There's a category called helical. There's a category called icosahedral. And a category called complex. These are shape categories. And these charts from your book are kind of showing under these purple lines what those shapes look like. So both of these would be helical. This is an icosahedral. These are icosahedral. And this is a complex. So I kind of want to draw what the shape categories are. So a helical virus is either going to be looking kind of straight like that, a straight line, Or it might be that same kind of shape, but kind of coiled. Both of those could be considered a helical virus. If a virus is icosahedral, it will kind of have a geometric looking shape. I'm not very good at drawing those quickly, but if it looks like kind of a blob, that's icosahedral. And then if it's complex, it's going to look something like that, where it's got both parts together. So a complex is got an icosahedral part plus a helical part together in one virus, and then it may have some other parts also. I have a doll of one of these in my office. Come see it sometime. Complex virus. Okay, so those are the general shapes. Viruses can have 5 kinds of components or structural components. And these are a list I want you to memorize. This is something on exam 2. List the viral components and tell which ones are required for all viruses. So this is like a section. What are viruses made out of? So I'm going to draw this a little bit separate. I'm going to come back to that picture in just a minute. No, I'll use it. Okay. So viral structures. Call this viral structures. All viruses have to have a genome. Or some kind of nucleic acid. Every virus has some kind of genetic information. It's absolutely required. And all viruses have to have a structure called a capsid. Or capsid proteins. These two are essential for every kind of virus that exists in the whole world. It's at least going to have genes, and it's going to have capsid proteins. The point of the capsid protein is to surround the genome and protect the genes from damage. So you've got gene information and then some proteins to protect that. Some viruses, that's all that they are. That's it. Just those 2 parts. And then there are 3 other structures that are possible. Third structure is called envelope. Some viruses would have this structure called envelope. And what that is, it's a membrane around the capsid. If they've got that, they got the envelope from the host cell. Parts 4 and 5 I'll come to in a second. So viruses might be as simple as just having the genome and the capsid. And if they've got an envelope, we call their category enveloped. If they don't have an envelope, we call their category naked. So the opposite of an enveloped virus is a naked virus. So here would be one, you've got the nucleic acid, It's got capsids around it. It looks like a coffee straw. It's naked because it doesn't have a membrane. This one, it's got nucleic acid on the inside, capsids surrounding it. It's got twisted helix, and then it's got this membrane around it, envelope. So it would be enveloped helical. So if the virus does not have an envelope, we say that that's a naked virus. I guess you could call it naked, but just go ahead and call it naked virus. Okay. The fourth thing that some viruses will have is called spike protein. So here's an example. Spike protein. Spike proteins aren't to protect your genes. Spike proteins stick out and allow binding to the host. They're going to stick out from the rest of the capsid and kind of be dangling on the outside, and they're going to control binding to the host. Yeah.
Where does that word say after the membrane capsid?
Comes from host cell.
Sorry, I just can't see. I don't know.
Yeah. I'm trying to get it visible on the Teams camera, which is why I quit writing on that side. Yeah. If you've got an envelope, it comes from the host cell, and we'll see how that happens in a few minutes. Spike proteins stick out and allow binding to the host. And then component 5, I don't have a picture of that one. Some viruses are going to have enzymes that they're carrying along with them as part of their structure. Those are more rare. Every virus is going to have a genome. Every virus is going to have capsid protein. Some viruses have these. Not every virus has these, but something like coronavirus. It's got genome, it's got capsid. It does have an envelope. It does have spikes. It does not carry enzymes. So to be coronavirus, it has to have all of its usual things. Tobacco mosaic virus, which is this one that looks like the coffee straw, it doesn't have an envelope. It doesn't have spikes. It doesn't have enzymes. It's just part of its nature that it only has genome and capsid. You can have spike proteins and an envelope. If you do, here's the membrane. Here's the spike protein sticking out through the membrane. Coronavirus is one of those. So those are the 5 parts. In this space over here under the nucleic acids, viruses can have weird genomes. Their genomes can be DNA, double or single-stranded. Their genomes can be RNA, plus or minus stranded, and that's like the plus stranded would be, it acts like an mRNA in the cytoplasm. Minus stranded is it could not be acting like a messenger RNA. So their genome types can be really different than ours. Coronavirus is an RNA genome. We'll have a separate day about that later, but which one of these, so what's our genome type? DNA, double or single? Double. So that's what we like take for granted. That's what genomes are like. Well, viruses can have other weird kind of genomes. And their genome determines what category they're in. Here's a picture of what spike proteins would normally be doing. So here's the virus. Here's some proteins sticking out of the surface of it, spikes. And see how the spike is matching? Alyssa Talabat, can you turn off the front light switch? Thank you. Okay, that's better. See how the shape of this spike is matching this host protein? That means that this virus can infect this host because the spike matches something on the host cell. Delta variant binds tighter to our host cells, so it's better at infecting. That's what makes it better. And it's like 3 or 4 amino acids that happen to match better, so it's more infectious. We'll talk about that next, either Friday or next week. So the spike protein controls what kind of cell a virus can get into, what can it infect. This is so small, it's not even useful. So what I want to start on next is what does a virus have to go through to actually infect? It's this stuff from your book, but I'm going to write about each step separately and draw it. This is also a list of 5 things that you need to memorize these steps. So you've got 5 viral structures. Genomes and capsid, all viruses have to have that. The other 3 are optional. And then we have 5 stages in viral life cycle. And we're going to start a drawing. And we'll use this drawing today and next time to show all of these steps. Okay, so this is going to be human cell. Here's our nucleus of our human cell. And our human cell has some receptor proteins on its surface. And out here we have a virus, icosahedral. And it's got spikes sticking out. That's the start of our picture. And notice kind of that these spikes match the shape of the receptor on this host cell. So this is our human host cell over here. So the first thing that has to happen for a virus to actually infect a host cell is it has to do something called adsorption, adsorption, add with a D, adsorption. Or some books would call this attachment. Those mean the same thing. Adsorption or attachment, that's step 1. if that's going to happen, you have to have the spike matching the host cell receptor, and they have to bind to each other so that the virus sticks there. If there's not a shape match, the virus can't infect that human cell. That's adsorption or attachment. So for that to happen, virus must stay bound to the host receptor. Okay, step 2, right over here. Step 2 is penetration step. And during this step, at least the genome of the virus, or let's say at least the viral genome, must enter the host cell. Sometimes it's just the genome, but sometimes it might be the whole virus gets into the host cell. So if this virus were to go through penetration step 2, the virus is now in the host cell. There's different ways that it could actually get in there. We'll talk about that next time, but that's step 2. The virus has to be, at least the genome, get into the host cell. And you can see the cartoon of this here. So this virus, it's like the host cell is engulfing it and bringing it in. The whole virus comes in. Some viruses act like that. If the virus has a complicated shell around it, it's going to do something called uncoating. That step some viruses do and some don't. So some viruses do uncoating step in the host cytoplasm. But overall, that's that stage of getting the virus, or at least the genome, into the host cell where it can actually do something. That's called penetration step. Next 3 steps, we'll get to that next time. So stay with these slides. And this also includes slides for influenza, which I think is the Friday topic. So we'll keep going with those Friday.